Aside from plant hormones, there are also other chemicals that can induce germination in seeds. In this lecture, we will look at a few known chemical promoters of seed germination, specifically hydrogen peroxide, potassium nitrate, and thiourea. Let's start with hydrogen peroxide. Hydrogen peroxide is a form of reactive oxygen species, or ROS, that is naturally produced in plant systems. ROS are oxygen molecules that gain electrons. Examples of ROS species are ozone, hydrogen oxide, hydrogen dioxide, and of course hydrogen peroxide. As I mentioned earlier, ROS, which includes hydrogen peroxide, are natural byproducts of physiological and developmental processes. Examples of these processes are photosynthesis, which occurs in the chloroplast. So you can see the generation of hydrogen peroxide here. We have photorespiration occurring in the mitochondria and beta oxidation occurring in the peroxisome. If you will recall, beta oxidation is a cyclic process that breaks down lipids. At lower concentrations, hydrogen peroxide can act as signaling molecules that regulate plant responses to stress. This slide illustrates how hydrogen peroxide can regulate stress responses in plants. When plants are subjected to environmental stresses like drought or heat, it produces hydrogen peroxide at low concentrations, particularly in the roots. The stress that we are talking about here is just mild stress, when the plant is not even showing signs of that stress. The hydrogen peroxide, as a signaling molecule, travels from the roots to the leaves and signals the stomata to close to avoid further loss of water. Essentially, it functions like abscisic acid or ABA. But the difference is that hydrogen peroxide acts very early on at the onset of stress. When the stress is severe, then a combination of signaling molecules, which includes ABA, acts in concert in an effort to regulate water loss in the plant. With the generation of hydrogen peroxide, the plant also produces antioxidants, which are responsible in the scavenging of hydrogen peroxide and ensuring that it doesn't accumulate in the cells. So how does the antioxidants regulate hydrogen peroxide concentration? Hydrogen peroxide is unstable and therefore very reactive towards macromolecules. The antioxidant's job is to donate electrons to the unstable hydrogen peroxide to make it stable and therefore non-reactive. At high concentrations, hydrogen peroxide becomes toxic. It can cause oxidative stress to macromolecules such as DNA, proteins, as well as lipids. In fruits such as tomatoes, this oxidative stress manifests into the gradual rotting of the fruit. At the cellular level, this is what's happening. We have here a healthy normal cell. When ROS, which includes hydrogen peroxide, accumulate, they start attacking the cell. That means ROS starts breaking down the lipids in the membranes, the DNA, and the proteins. With macromolecular degradation, the cell suffers from oxidative stress until it dies. During germination, hydrogen peroxide at low concentrations has important functions in the seed. For one, it serves as a signaling molecule that regulates the balance of phytohormones in the seeds. It is also responsible for the selective mRNA and protein oxidation, which is important for the mobilization of stored proteins to the embryo axis. Hydrogen peroxide also aids in facilitating seed germination by releasing dormancy, weakening the endosperm, inducing programmed cell death of the alurone layer, as well as acting as a disinfectant that protects the seeds from pathogens. Hydrogen peroxide has been used to stimulate the germination of conifers, legumes, tomatoes, and barley. Specifically for conifers, hydrogen peroxide has been used as a rapid viability check. Hydrogen peroxide can induce germination as it acts as a respiration stimulant that accelerates the breakdown of food reserves in the seed. The faster breakdown of stored reserves provide faster supply of energy and materials needed by the embryo axis to develop. Additionally, hydrogen peroxide has disinfectant properties that prevents the growth of molds on the seeds or in the germination media. This slide shows you the effects of hydrogen peroxide treatment on the germination of pea seeds. We have here germinating seeds at 6, 24, and 48 hours of water uptake following treatment with 0, 20, 40, and 80 millimolars of hydrogen peroxide. After 6 hours, it is clear that seeds treated with hydrogen peroxide germinated faster compared to the non-treated seed. These are the treated seeds, and this is the non-treated seed. At 24 and 48 hours, you can see that the radicle, or the embryonic root, as well as the shoot coming out of the seeds treated with 20 and 40 millimolars of hydrogen peroxide are longer compared to seeds treated with 80 millimolars of hydrogen peroxide, as well as that of the untreated control. Seeds of peas treated with 80 millimolars of hydrogen peroxide also exhibit abnormal curling of the root, as you can see here, and also here. So what does this tell us? This data shows that hydrogen peroxide, when applied at lower concentrations, is able to improve germination. But at higher concentrations, it can be toxic to the seed, resulting in abnormal root growth. Aside from hydrogen peroxide, potassium nitrate is another chemical that has been used so often to induce better germination in seeds. Potassium nitrate is more commonly known as saltpeter. Most seeds that are sensitive to light are also sensitive to potassium nitrate. Solutions of 0.1 to 0.2% potassium nitrate are commonly used in routine germination testing. Potassium nitrate has been reported to interact synergistically with temperature and phytohormones such as gibberellic acid and cytokinin to induce germination. Gibberellic acid and cytokinin, as you already know, are positive regulators of germination. Potassium nitrate has also been known to reduce ABA content in the seed to promote germination. ABA is a known germination inhibitor. Additionally, potassium nitrate has been reported to weaken the seed coat and make the seeds more permeable to water and air. From previous lectures, you should already know that water and air are necessary requirements for seeds to germinate. Potassium nitrate treatment helps the seed take in water and air by softening the seed coat and allowing both water and air to pass through. This slide illustrates the effects of potassium nitrate in the germination of Allen-Rolfia occidentalis. Allen-Rolfia occidentalis is a low-lying shrub found in southwestern U.S. and North Mexico. It grows in sandy, often salty alkaline soils, such as desert washes and dry saline lakebeds. These graphs illustrate the effects of potassium nitrate in the germination of Allen-Rolfia occidentalis seeds. The graph on the left shows germination of the species when supplied with varying concentrations of NaCl or salt, but without potassium nitrate. The graph on the right shows the germination of the species when supplied with varying concentrations of salt with the addition of potassium nitrate applied at 20 millimolars. Salt was applied at 0, 200, 400, 600, and 800 millimolars. Now, if we compare the two datasets, it's clear that potassium nitrate application allows better germination of seeds, especially if we look at the germination of seeds supplied with 400 to 800 millimolars of salt. This line indicates the germination of seeds treated with 400 millimolars of salt. Without potassium nitrate, the maximum germination was only around 60%. In seeds treated with 600 to 800 millimolars of salt, as indicated by these two lines with the the diamonds and the triangles, germination did not even reach 10%. Again, this is without potassium nitrate. But with the addition of potassium nitrate in this graph, you can see that germination reached more than 85% when treated with 400 to 800 millimolars of salt, as indicated by the lines with the squares, right here, the diamonds, as well as the triangles. Then we have thiourea. Thiourea is a water-soluble organosulfur compound that is structurally similar to urea, except for the presence of a sulfur atom in place of oxygen. Seeds that have specific requirements for light and temperature can be treated with thiourea to meet these exogenous requirements. Thiourea promotes germination by inducing the buildup of gibberellin and cytokinin, especially in seeds that require stratification. As you already know, gibberellin and cytokinin are known positive regulators of germination. This slide illustrates the effects of thiourea in the germination of Allenrothia occidentalis seeds. The graph on the left shows germination of the species when supplied with varying concentrations of NaCl or salt, but without thiourea. The graph on the right shows the germination of the species supplied with varying concentrations of salt in combination with thiourea applied at 10 millimolars. The salt was applied again at 0, 200, 400, 600, or 800 millimolars. Now, if we compare the two data sets, it's clear that thiourea application allows better germination of seeds, especially if we look at the germination of seeds supplied with 400 to 800 millimolars of salt. This line indicates a germination of seeds treated with 400 millimolars of salt. Without thiourea, maximum germination was only around 60%. In seeds treated with 600 to 800 millimolars of salt, as indicated by the lines with the diamonds and triangles, these two lines right here, the germination did not even reach 10%. And again, this is without thiourea. But with application of thiourea, you can see that germination reached about 65 to 70% when treated with 600 to 800 millimolars of salt, and up to 100% in seeds treated with 400 millimolars of salt. In summary, other chemical compounds such as hydrogen peroxide, potassium nitrate, and thiourea can also be used to promote seed germination. Hydrogen peroxide is a naturally occurring reactive oxygen species that can stimulate germination at low concentrations by weakening the endosperm, accelerating the breakdown of stored reserves, and releasing dormancy in seeds. Potassium nitrate promotes germination by reducing ABA content and weakening the seed coat to make the seed more permeable to water and air. Thiourea promotes germination by substituting for the light and temperature requirements of germination. In seeds requiring stratification, thiourea promotes germination by facilitating the buildup of gibberellin and cytokinin, which are known positive regulators of germination.